welcome to my channel Sam Bios Plus. Friends, today we'll come to know about the key similarities and differences between scanning electron microscope and transmission electron microscope. But before that, will you please consider subscribing my YouTube channel? Your subscription and viewership are often a lot to me. Scanning electron microscopy (SEM) and transmission electron microscopy (TEM) are two distinct electron microscopy techniques that are used to study different types of samples on the nanoscale. First of all, let us know about the similarities. Number one, electron source. Both SEM and TEM use an electron gun to generate high-energy electrons, which are then shaped into a beam. This electron beam is then used to illuminate the sample. Number two, electromagnetic lenses. Both of them requires different types of electromagnetic lenses to focus, shape and control the trajectory of the electron beam. Transmission electron microscope use three types of electromagnetic lenses, such as condenser, objective and projector. However, in SEM, projector is absent but remaining two are present. In addition to these lenses, apertures are also used in both. Number three, vacuum environment. In both SEM and TEM, vacuum environment is essential. My question is that what will happen if vacuum is not maintained? In the absence of vacuum, electrons will collide with air molecules that will result in image distortions. In addition to this, there will be electrical discharge and arcing that will damage the electron source. And the atmospheric moisture that is present in the microscope column will quickly ruin the sample, thereby rendering the image formation impossible. Number four, image formation. In both SEM and TEM, image is formed based on how the electrons interact with the sample. In TEM, image is formed from the transmitted electrons. By contrast, in SEM, backscattered electrons and secondary electrons are used to generate the image. In addition to this, both of them can be equipped with detectors such as energy dispersive X-ray spectroscopy to analyze the chemical composition of the sample. Now differences. Number one, how it works. Scanning electron microscope with the help of scanning coils sweeps a beam of electrons across the sample surface in a systematic line-by-line rectangular pattern. Such type of scanning is known as raster scanning, which generates two types of signals. Number one, secondary electrons, and number two, backscattered electrons. Backscattered electrons are the electrons of the original beam that are bounced off the sample. These electrons are of high energy and are used to study the chemical composition of the sample. On the other hand, secondary electrons, which are of low energy, are ejected from the sample surface. As these electrons are ejected from the top surface layer of the sample, they are used to study the surface topography. On the other hand, in transmission electron microscope, A beam of fast-moving electrons is transmitted through an ultra-thin sample. Only the transmitted electrons which pass through the sample are captured by the detector to form the image. Number 2. Primary use. SEM is used to study the surface topography and chemical composition of different types of samples. By contrast, TM is used to study the internal structure of different types of biological samples and also to study crystal structure, lattice defects and atomic level imaging. Number 3. Image type. SEM generates a three-dimensional image. On the other hand, the image which is generated by transmission electron microscope is two-dimensional. It is similar to the image generated by the traditional bright field light microscope. Number 4. Sample preparation. For SEM, sample preparation is minimal. By contrast, in case of transmission electron microscope, sample preparation is highly complex. In this case, different types of samples are cut into ultra-thin sections by ultramicrotome. These sections are then stained with uranyl or lead acetate to improve the contrast. Number 5. Resolution. The resolution of SEM is less than that of TM. Its resolution is about from 1 to 5 nanometer. On the other hand, the resolution of TM is extremely high and it is about 0.1 nanometer. Number 6. Magnification. The magnification of SEM is less than that of TM. Its magnification is determined by the ratio of the displayed image size to the area of the specimen that is actually scanned. Its magnification is from 1 to 2 million times. On the other hand, the magnification of TM is very high. It can magnify the specimen up to 50 million times. The magnification of transmission electron microscope is primarily determined by the current that is applied to the lenses. In such instrument, there are two lenses that contribute to the total magnification. First of all, objective creates an intermediate magnified image which is then further magnified by the projector to form the final image. So friends, this is all about the similarities and differences between SEM and TM. If this video is really useful, like and share with your friends and also subscribe my YouTube channel.